This video presents the general features of the hardware and software, as well as sample handling procedures for the dispersion technology instruments. These instruments are designed for characterizing concentrated dispersions and emulsions without the need for dilution. There is also an option for characterizing some properties of porous materials. What you are looking at now is the most general setup of the DT1202, which contains all options. This is our lab unit, which has been through years of use, so please disregard any wear or signs of aging. There is an electronics box that contains all electronic components, as well as the computer hardware and software used for conducting all of the measurements. The back panel of the electronics box has a set of outlets for connecting the cables of various probes that are placed into the measuring unit stand. Each outlet and cable are unique, which eliminates the possibility of plugging the wrong cable into the wrong outlet. The set of probes includes acoustic sensor for particle sizing and high-frequency longitudinal rheology, zeta potential probe, temperature probe, pH probe, conductivity probe aqueous, conductivity probe non-aqueous, heater probe, and burettes for titration. You can find a detailed description of the probes, their specification ranges, and precision levels on our website, or in the brochures that can be downloaded from the website. Dispersion Technologies instruments come in various configurations and with various probes. Different setups are given different model numbers. What we have here is a DT1202, which is designed to measure both particle size and zeta potential. Instruments for solely particle size measurements are in the DT100 model line, and instruments which measure only zeta potential are in the DT300 model line. However, different probes and options, including those I mentioned earlier, can be included with any of those models. Decades of experience dealing with concentrated samples has led us to develop a variety of sample handling setups to better accommodate the wide variety of concentrated samples the DT instrument can measure. The setup that you can see now is suitable for rather dilute, low viscosity samples. The magnetic mixer that is placed on the bottom of the sample chamber cannot stir heavier, more concentrated samples. For more viscous samples, we can use a peristaltic pump. Mixing is required only for samples that settle or for titrations. Otherwise, we can work with stable, stationary samples, which allows us to minimize sample volume and simplifies cleaning. It is convenient in such cases to work with different sensors individually. Minimization of the sample volume for acoustic particle sizing is achieved by using this insert, which makes sample volume around 15 milliliters. Alternatively, we can use the acoustic sensor as a standalone sensor with an even smaller sample volume, around 3 milliliters. Minimizing sample volume for the zeta potential probe can be achieved using these cups, which are placed on the face of the probe. Minimum sample volume is 0.1 milliliters in the case with the steel cup. The zeta potential probe can also be inserted into the sample in an external vessel, which can be stirred or not. We also have a special setup for the zeta potential probe for pumping sample through with a peristaltic pump. The injection probe for the titration burettes can be inserted into the sample as well, which would allow for conducting titrations of heavy viscous samples. When characterizing the zeta potential in porous materials, the probe can simply be placed in contact with a porous block, as we will demonstrate now. We also have a special setup for measuring the porosity of porous material with our aqueous conductivity probe, which functions at a high frequency of 3 MHz in order to eliminate electrode polarization. Now I will demonstrate general features of the software, and will set up a measurement of our standard reference material, silica ludox. This is precipitated silica at 10 weight percent with particle size of 30 nanometers and a zeta potential of -38 millivolts. This is the Dispersion Technology proprietary software. This program allows you to operate all of the sensors. This section is for defining your sample. These two combo boxes are linked to a material database. I just have to select a material for the medium and one for the particles. The Ludox sample is water-based. That is why I selected water for the medium and silica Ludox as the material for the particles. Now I should define what it is I want to measure. This section allows me to design my experiment, which consists of one or many measurements. In order to measure particle size, zeta potential, and conductivity, I should check these boxes. I can have the instrument conduct multiple measurements if I'd like. Now the instrument will make 3 consecutive measurements of particle size, zeta potential, and conductivity of the silica Ludox sample. The values of pH and temperature are measured every 2 seconds and displayed on this frame. You can also define various titrations using this form. There are 5 types of titrations: pH stat, pH ramp, milliliter titration, milliliter equivalence titration, and time titration. Demonstrations of these titrations will be done in a separate video. Samples are loaded by simply pouring from the top. If I want to mix the sample, then I turn the mixer on. A rotating, magnetic, pill-shaped stir bar on the bottom of the sample chamber mixes the sample during measurement. It not only provides vortex mixing, but also recirculates sample from the bottom of the chamber back up to the top via this tube. This helps prevent settling if large particles are present. Now I click on the Run button and the experiment starts. Measurement of particle size usually takes around 5 minutes. Zeta potential, on the other hand, takes less than 1 minute. After a measurement is finished, the software displays the analysis form, which calculates all parameters and reports results of the last measurement. This form contains 2 graphs. One graph represents particle size distribution, and the other one shows the raw data, which is the ultrasound attenuation frequency spectra. This second graph is important for judging the reliability of the data. It displays experimental points in dark blue, as well as the theoretical fit. These light green points correspond to this particle size distribution. You can see that the theoretical fit fits the experiment very well, which supports the reliability of this particle size distribution. These boxes display the cumulative values of the particle size distribution. Values of the zeta potential and Debye length are shown on these boxes. You can store all of these results to Excel using this menu item. All data is automatically stored to the database. You can select various experiments and recalculate them, or overlay them using these buttons. Database selections can be exported to Excel files as well. More detailed descriptions of the instrument functions are presented in the manual, which can be accessed directly from the home page. We hope that you have enjoyed this short tutorial, and thank you for your attention.